Okay. Um, so today we're talking about the humoral immune system. Um, Continuation from last week. Remember that the humoral immune system are the humors, the things that are in the plasma that could be transferred from one person to another um, to help with immunity, which we now understand to be mediated by antibody. And the other component is complement that we're going to talk about today at the end. So, very, very high yield information today for board exams. Okay, so antibodies, their function is to neutralize and eliminate microbes and toxins. As we know, they're produced by B cells slash plasma cells in the lymphoid organs, bone marrow, and they perform effector functions distant from where they are produced. So they actually get into the bloodstream and travel before they are active. They are derived from both short-lived and long-lived plasma cells following activation of either a naive or memory B cell. Their function is mediated by the heavy chain constant region, and the different heavy chain isotopes serve distinct functions. So the IgG, IgA, IgM, and IgE. And then the function is triggered by binding of the antigen to the variable region, which is both part of the heavy chain and light chain. So here are some of the jobs of antibodies. One, they can just neutralize microbes and toxins. Two, they will opsonize them or make them more prepared to be phagocytosed. Three, they will participate in antibody-dependent cellular cytotoxicity, otherwise known as ADCC. But then they can also are very important for complement activation, and the complement, as a result, can either lyse the microbe or can help with phagocytosis or just promote general inflammation. So here are the different isotypes: IgG, IgM, IgA, IgE, and then of course to a lesser extent, we know lesser about IgD. So the IgG has several functions. It's one of the main opsonizers for phagocytosis by macrophages and neutrophils. It activates the classical pathway of complement. IgG3, most potent, then IgG1, and then IgG2. IgG is essential for antibody-dependent cell-mediated cytotoxicity. And then at the very end, we're going to talk about neonatal immunity. IgG is the isotope that gets transferred from mom across the placenta and gut. IgM also activates complement, and it is the antigen receptor on naive B cells, like we talked about last week. IgA is essential in mucosal immunity. It's secreted both across the entire GI tract and respiratory tract. Um, and it can activate complement by the lectin or alternative pathway. And then IgE is essential for mast cell degranulation and plays a role in immediate hypersensitivity. Okay, neutralization antibody can block the binding of microbes and toxins to cell receptors. This is direct steric hindrance. In the blood, this is primarily done by IgG. In the mucosa, it's primarily done by IgA. So, direct steric hindrance is it's just binding so that there is no room actually or availability for this microbe to actually then bind onto the receptor. So it's just physically blocking it from being able to do its job. It will also do this with toxins. It's just not microbes, but it can do the same thing with toxins. Second job, opsonization and phagocytosis. So IgG will coat microbes and then it promotes phagocytosis by binding the Fc receptors on phagocytes. So FC receptors will then promote phagocytosis of the opsonized particles and deliver signals that stimulate microbial activities of the leukocytes. This is done with the FC gamma receptor. So here are the different antibody receptors. Notice that, like all receptors, it has this extracellular domain, transmembrane domain, intracellular domain. And then, just like before, it has these ITAMs, which are stimulatory, and ITIMs, which here are in blue, which are inhibitory. The primary inhibitory antibody receptor is the FC gamma R2B. Since that's the only one, I think that's something that I would commit to memory. The nomenclature is written here because the FC portion of the gamma or IgG, or in this case, alpha, FC alpha, or FC epsilon, it just tells us what is binding to the receptor, R for receptor, and then they're just named in order, R1, R2, R3. In general, R1 means it has the highest affinity, R3 is the weakest affinity. So FC epsilon R1 binds the FC part of IgE, and R1, this is the strongest FC receptor. You can see that it has an alpha chain, two gamma chains, a beta chain, and it has ITIMs on the beta and gamma chains. Let's skip to the FC epsilon R2B. It's just one receptor. It has this inhibitory, the ITIM on the intracellular part. FC gamma R2 is another example. It has alpha and two gammas. The ITIMs are on the gamma chains. Okay, so here is a nice picture of exactly what happens when opsonization occurs. So again, you have your microbe and you have your IgG antibody, but instead of acting just out of steric hindrance here, or just physically blocking this microbe from being able to bind to a receptor in order to affect a cell, it is purposefully causing this microbe to be more likely to bind to a receptor on a phagocyte, and specifically these FC gamma R1s. So as soon as it binds, then it will activate, it will send signals into the cell to not only phagocytose, but then to upregulate all the intracellular mechanisms of killing that we have discussed before. Okay. Next job of antibody is something called antibody-dependent cell-mediated cytotoxicity, or ADCC. What happens in this one is that NK cells will bind to an antibody-coated cell by its FC receptor and will destroy the opsonized cell. So instead of actually the microbe being opsonized, it is a cell, an infected cell, that is getting opsonized, and it gets killed by NK cells. This happens through the FC gamma R3 receptor. And here is a nice picture of exactly how this works. So here is your, here is your cell, which could be self, could be infected, and there's antigen getting displayed. IgG will bind to it, and through FC gamma R3 on the NK cell, it will bind, and that will signal the NK cell to actively kill that cell off. Okay. For IgE, its main job is to get rid of helminthic parasites. So antibodies, specifically IgE, mast cells and eosinophils will mediate the expulsion and killing of helminthic parasites. Phagocytes in general are unable to kill off helminthic parasites because they're large and they have what I kind of think of as a thick skin. It's just the mediators that a phagocyte will make, those reactive oxygen species, have a hard time actually breaking down that outside of these helminthic parasites. So we employ other things instead, like histamine in mast cells and major basic protein in eosinophils. And this signals through FC epsilon R1. So that high affinity IgE receptor IgE will bind to something, a helminthic parasite. It will bind to that Fc epsilon R1, and then in a mast cell, for example, it will release histamine, and then histamine can very effectively chew through that kind of thick skin of the helminthic parasite and be able to kill it off. So it's just slightly different microbe, just needs a slightly different method in order to destroy it. Okay. I'm going to pause there just for 1 second, see if anyone has any questions about antibody. If not, we'll move on to complement. Yeah, both that part and this part high yield for boards. That's what I can say. I think I would know every slide on here pretty well. Okay, so complement is a major effector of the humoral immunity. It consists of serum and cell surface proteins that interact to eliminate microbes in a cascading fashion. This cascade happens by activation of sequential proteolysis of different proteins to generate proteolytic enzyme complexes. So it's just a cascade where you get proteolysis and you get a new enzyme complex, and you get proteolysis and a new enzyme complex, and so on and so forth, to make several different effector functions. In general, the products will covalently attach to a microbe surface or antibody on a microbe surface. And is started through 3 distinct pathways: the classical, alternative, and lectin. Most of this chapter really deals on how this is activated, but it is equally important, in my opinion, to remember that there are certain regulatory proteins that will inhibit this whole system to prevent complement, for example, destroying normal host cells. Part of the reason I think this is very important to know is because clinically there are some diseases if you have a mutation or absence of some of these inhibitory regulatory proteins. Okay. So all these different pathways depend on generation of 2 complexes. And I think that is one of the most important things you can keep in mind when you're trying to study the complement system. You're trying to get a C3 convertase and you're trying to get a C5 convertase. One leads to the other. So no matter how you start the system, you're trying to get a C3 convertase because the job of a C3 convertase is to cleave C3. And once you have accomplished that, then you can make a C5 convertase with that cleaved C3. Excuse me, and that C5 convertase will cleave C5. Excuse me. So no matter how you start, it is important to know the differences in how it starts, but it is all going to come through a C3 convertase and a C5 convertase, which can be slightly different depending on how it starts. So here are examples of, or here is schematic of the alternative, classical, and lectin pathway. But as you can see, you have a major difference in how it starts, but then ultimately you're going to get a C3 convertase, even though it may be different, and that is going to cause cleavage of C3, which will lead to formation of a C5 convertase, which will then lead to a MAC complex being made to poke holes in the outside of the cell. So starting with the alternative pathway, this is when you actually just get spontaneous cleavage of C3. This happens millions of times in a minute in anyone's body. C3 is spontaneously cleaved to C3A and C3B. Remember, the reason this doesn't typically end up with just killing off your own cells is because we have these inhibitory proteins that we're going to look at before that are associated with mammalian cells. So in general, we should not have our complement system attacking ourselves. So once you get that spontaneous cleavage, if there happens to be a target cell around, in other words, a microbe, then that C3b will attach to its cell surface. If it does not, in less than a second, that C3b is inactivated. But if there happens to be a microbial target there, it will attach. And then something called factor B will bind to C3b. So then you have something called C3b. And then factor D cleaves factor B. So then you have cleaved factor B, which we call BB. So you have a combination of this C3b and then the BB added. And this is the alternative pathway C3 convertase. Don't let this overwhelm you. It's just a path. You get spontaneous C3A, C3B, and then you get cleavage C3B, add a B, it's cleaved, and you get this C3B, BB. It's alternative pathway convertase. Something called propyridine will stabilize C3B, BB. Propyridine happens to be the only known positive regulator of complement. Propyridine deficiency, I would look up, is an immune deficiency that you should be aware of. The attachment of propyridine is just another checks and balance. It's favored on microbes and should not happen on mammalian cells. And then a second C3b will bind to that already C3b, BB, or that C3 convertase. And that addition, after you get stabilization propyridine, you get a second C3b. That, remember, that was just spontaneously cleaved. The addition of that second becomes that alternative pathway C5 convertase. Excuse me. Okay. So here is a picture of all this happening. You get the spontaneous cleavage. Remember, you get almost immediate inactivation of C3b if it does not immediately see a microbial surface. So in a normal body, you'll get C3bb, which is nearly immediately inactivated. The byproduct of the spontaneous cleavage of C3 is C3a. This is something considered a nephrotoxin, or something that can mimic anaphylaxis. Okay, but if you have your C3b and there happens to be a microbe there, then it should bind to the surface. And then you have a B that binds to it. Factor D will cleave, and this is where you have your C3 convertase. Stabilized by propyridine, you get more C3 cleavage. These C3Bs will attach, and then you get a double C3B, BB, and that's your C5 convertase, which will start cleaving C5 to C5B, and then second anaphylatoxin called C5A. Okay, I'm going to take a quick pause. It's a lot of words, but I think really just understanding the movement that cascade from one to the other is the important part. Okay. Yeah, I don't know that there's much additional to take away from this table. These are obviously the essential components of the alternative pathway. I think the big thing to know is that propyridine is a stabilizing, and it's the only one that's something that's likely to come up. Okay, moving on is the classical pathway. So this is initiated by binding of C1, complement 1, to IgG or IgM. On IgG, it happens to be the heavy chain region 2, on IgM, region 3. C1 is made actually of 3 components. So C1q is a hexamer which binds to antibody. C1r is a dimer which is a protease and activates C1s. And C1s is a dimer which is a protease and cleaves C4. Okay. Don't get too confused yet, but this entire structure here in blue, yellow, orange is C1. It's actually a complex. And this is binding to these heavy chains um, on the IgG, which have already bound to the cell surface of a microbe. In this case, in this classical pathway, C4 is homologous to C3 in the alternative pathway. So once you have cleavage of C4 by C1, C2 will bind to that C4b, and then C2 is cleaved by C1s, and you get C4b, C2b, which is the classical pathway C3 convertase. Once you get C4b, C2b, it will generate C3b, C3 convertase, which will bind onto that complex already, and that makes the C5 convertase. The alternative pathway, that spontaneous cleavage of C3, can also amplify this process. But once you get that C3 convertase, you convert C3, more C3b, it binds, and you get a C5 convertase. Okay, so let's look at this. So here you have your antibodies bound to your microbe, and then this C1 complex, the hexamer part, will actually bind then to the antibody. And then C4 will bind to the C1q, and then the C1s will bind that, will cleave that C4. So you get a C4b. You can get a C2 that binds, and then you'll get cleavage of that C2. So you have C4b, C2b, and that is your C3 convertase. You then will start cleaving C3 with that C3 convertase, and you make more C3b that can bind. And then once you have all three of those components, you get a C5 convertase. And then the early stages, that's all we're trying to do, is to get to a C5 convertase and start making C5b. Okay. So IgG and IgM are the two types of antibody that can activate complement. IgG, preferentially IgG3, then IgG1, and then IgG2. I remember this because of the area code of Chicago. It's 312. So I always remember that. It is worth noting that soluble IgM cannot bind complement. It has to actually be bound for it to be able to bind complement. Same with IgG, that IgG first needs to be bound to the microbe for that complement to work. So you should not get complexing of just your antibody and C1 in your blood. It's another checks and balance. It should be that this is binding to a microbe for the system to take off and start working. Okay, so here are all the different components. You have your C1 complex, which is QRS. Q is the part that binds to the antibody. C1R cleaves C1s, and C1s cleaves C4 and C2. Okay. Finally, the last pathway is the lectin pathway. So this starts by binding of a microbial polysaccharide to a lectin. An example is when there's mannose on a microbe, then there's something called a mannose-binding lectin that will initiate this pathway. There is an immune deficiency associated with mannose-binding lectin. There is no treatment for that here in the United States, but my understanding is there's treatment in Europe for mannose-binding lectin deficiency. So again, I think that's worth knowing. So the lectins are homologous to C1q, that hexamer that binds the antibody, And then MBL will bind to MASPs. So MASPs are homologous to the C1R and C1S. And then MBL is associated with the serine proteases. So let's take a look at this one. So all the way over on the right here, you have your lectin pathway, and this mannose is homologous to the C1Q, and then you have your MAS1 and MAS2, which is homologous to your C1R and C1S. And then the same thing happens that you're initially cleaving C4 to C4B, and then you get a binding of C2, which is cleaved again, and you have a C4B, 2B. This is your C3 convertase, and the lectin pathway is exactly the same as the classical pathway. Once you have a C3 convertase, you can start cleaving C3 to more C3B. And once you add a C3B to that same complex, you have a C5 convertase. And remember, all we want to do is make C5 at this stage of the game. So this can happen three different ways, either spontaneously or two very similar kind of analogous ways, the classical and lectin pathways. Any questions there? Okay. There is one variant to bring to your attention. There are Sign R1 is a C-type lectin that will recognize pneumococcal polysaccharides, but also bind C1Q. This is found in the splenic marginal zone macrophages, and it can activate the classical pathway. Okay, so remember, all of this was to form a C5 convertase, and then we get to the terminal complement. Remember, terminal complement deficiency is associated, if there's a mutation associated with immune deficiency, that namely predispositions you to Neisseria infections. So once you have a C5 convertase, of course, its job is going to be to cleave C5 to C5b. C5b will then bind to C6 and C7, and then that C5b, C6, C7 complex will insert itself into a cell and become a receptor for C8, and then eventually many C9s is added on that complex to form a channel into the cell, or actually physically punch a hole into the cell. This allows movement of water and ions, so it just destroys the cell by letting everything that it needs to remain viable out. So here is a very nice picture. You have your C5 convertase making C5 into C5b. Once you have C5b, it binds to C6, C7, and C8. This punches into the cell, and then you get poly9s. So you get several poly C9s. So you get several C9s, and each one of these punches a hole. This is the membrane attack complex. The cell lysis is not because of one hole, but remember, this complement is activating all over the place. So you're probably getting thousands or millions of different little holes punched all throughout the cell. That's going to allow this gradient for water and ions to be exchanged in a way that makes that cell not viable anymore. Okay. Here are all the different components of late complement. I don't think there's much else to be said about those. Okay, now the complement receptor. These I think are just very hard. I think these are very hard to remember because in the case of complement receptor, this is one example where I know in immunology everyone wants to switch over to CDs, but we see all the time CR1, CR2, CR3 used interchangeably with the CD. So in this case, very unfortunately, I really do think you need to know that CR1 is CD35, CR2 is CD21, and so on and so forth. Okay. So complement receptor 1 will bind to C3b and C4b. and it will promote phagocytosis of C3b and C4b coated particles, and it will clear immune complexes from circulation. CR2 is also CD21. It stimulates humoral immune responses, promotes trapping of antigen antibody in germinal centers. It binds to C3d. And very importantly, you should know that is the cell surface receptor for EBV. Now I'm going to draw your attention to CR3, complement receptor 3 and complement receptor 4 at the same time. And as you will see, these are both integrins and they both have CD18 on them. So if you are missing CD18, we know you have leukocyte adhesion defect because you have no CD18, so you cannot adhere, cannot get those cells out to the periphery. So you have that immune deficiency, LAD. You have also lost two different complement receptors, CR3 and CR4. Okay, last two, which I certainly know less about, is CR immunoglobulin, which is on Kupffer cells and clears blood-borne pathogens, and then SINE R1 are just other examples. Here, let's see. There's a nice table of all these complement receptors. Right, I would remember these as cell adhesion, CD18 being part. Receptor for EBV, I think, is important. Knowing CD21 as the co-receptor for B cells. Of course, that B cell receptor complex is important to know. Complement receptor 1 and phagocytosis is probably what you need to know. But I don't know, I bet 2, 3, 4 are more important since they do have a little bit of nuance to them. Certainly, anytime there's a pathology, I know I've said this a million times, anytime there's a pathology associated with what we're learning, it's just something that you really should know, both clinically and for testing purposes. Okay, so let's step back a minute and talk about regulation of this whole system. So remember I said this happens when there is a microbial cell present. If it's mammalian cells, then it is unlikely to progress on and that cascade to happen. There are things that turn that off. So remember MASPs and the lectin pathway are analogous to CR1R and C1s in the classic pathway. Those are both inhibited by C1 inhibitor, which makes sense, inhibitor of C1. If C3b is on a mammalian cell accidentally, it will be bound by proteins that competitively inhibit formation of C3 convertase, including membrane cofactor proteins, CR1, DAF, and factor H. Here is basically that happening. Don't want it to become proteolytically activated. Okay, hold on. All right, let's step back. I'm not sure I love the order that I put this in, but C1 inhibitor will prevent C1R2 and RS from becoming proteolytically activated. But clinically, that's not when we use C1 inhibitor. And why is that? And why, when we're talking about angioedema, do we look at both this complement inhibitor as well as C4 to decide if someone may have hereditary angioedema? The answer is the same redundancy that happens all over the place. So it just so happens, I think I like maybe this one better. The C1 inhibitor is named because it was first discovered as an inhibitor of the complement pathway, but it has many jobs, and among them is this role in making bradykinin, plasmin, and the coagulation system into fibrin. The one that is important to us is this bradykinin system. So if you have no C1 inhibitor, you're going to make too much bradykinin. You're going to be predispositioned to have angioedema. So why do we check C4 levels? Well, remember, if you have no C1 inhibitor to turn off that C1 is going to keep splicing C4 to C4A and C4B. So you're going to just use it all up. So it's just a biomarker, and in and of itself is, to my knowledge, I think to all of our knowledge, it's not related to actually the process of angioedema, but but we can just exploit it by its other process. Because if it's not there, your C4 levels can go real low. There's no inhibition. It's just going to keep using it up, keep using it up, keep using it up. So if it's not there to turn off the complement system or this classical cascade of the complement system, it means it's also not there to stop this bradykinin formation, and therefore you have angioedema. Okay, that was an aside. I'm stepping back now to these inhibitors. So here is an example of, looks like DAF, MCP, and CR1, which will displace C2B from C4B. So remember, C4 is that first one that will actually attach to the membrane. And you have these competitive inhibitors, which instead of allowing this to become a C3 convertase, we'll just pop it off and it will inhibit this whole formation. This is showing both in the classical pathway, but also in the alternative pathway as well. Oops. Okay. So C3b is degraded by factor I, and MAC formation is inhibited by both CD59 and factor S. Here is CD59. It's not allowing the this poly C9, and factor S is not allowing the membrane insertion of C5b to C7. Okay. DAF and CD59 deficiency are both associated with PNH. So all of these, I think, are important to remember, but when they're attached to a pathology, they're very important to remember. C1 inhibitor, as we discussed, is associated with hereditary angioedema. So do I think you need to know any level of detail of any of these inhibitors? I really don't. But probably, definitely when they are, yeah, I would say definitely when they're associated with a pathology, that's probably what I'd know. And just being able to recognize them as part of the complement system in your brain, marry it to the complement system, and marry it as an inhibitor. Remember, don't confuse it with propyridine, which is the one stabilizing, activating component. The rest of these are inhibitors. Okay. Here is a summary of a lot of the different proteins that we just discussed. And then I added onto here diseases associated with different parts of the complement pathway, probably all of which you should know. Certainly with an increased frequency of infection, like Neisseria and these terminal pathways. But I'll upload this into the box folder so you can take a look at this table. Okay, so this is just a summary of all the different things that can happen from the complement system. One, I'm going to start at the bottom. Is the complement-mediated cytolysis, where you actually are just punching holes from these MACs into the cell. The other is you can just get stimulation of inflammatory response. These are from these little pieces, these anaphylatoxins C3A and C5A that get kind of discarded when C3 is turned into C3B. This will both recruit and activate leukocytes, and cause it upregulates the ability of leukocytes to destroy microbes. And then there is direct opsonization. Again, this is process of opsonization. It's basically just decorating the outside of these microbes to make them more recognizable to phagocytes. So same thing what we talked about with antibody can opsonize. C3b or C4b can both opsonize as well. Okay, I think this is the last concept that I want to touch on. There's really only one point here that I think is important, but there's something called the neonatal Fc receptor. It's a salvage from IgG from endocytosis. And it has a greater half-life in the blood compared to other isotopes. Now, why is this important? It is important because we have learned to exploit this and use it on several drugs to increase the half-life. And here is a list of different biologic therapies that have been, we've been able to prolong the half-life or the amount of time between administering these, which is ideal because most of these are some sort of injection and you don't want that with any frequency at all. But the longer you can make that, the easier to sell it is for our patients. So this is how we use that neonatal Fc receptor clinically. It just extends that half-life and probably a good fact to know.